Hi everyone, my name is Dr. Bonnie Quigley. I'm a microbiologist at the University of the Sunshine Coast here in Queensland, Australia.
Hi, I'm Dr. Andreas Kupz. I'm also a microbiologist and I'm based at the Australian Institute of Tropical Health and Medicine at James Cook University in Cairns.
As a microbiologist, I study viruses and bacteria and how they interact with people and animals. Most days, I'm trying to help koalas deal with the bacteria, chlamydia, and the virus koala retrovirus by studying these microbes and developing vaccines against them.
My research focuses on infectious diseases caused by bacteria and parasites. I'm interested in developing a vaccine against tuberculosis. Tuberculosis is a disease caused by bacterium, and it's transmitted when people cough up little droplets that we also call aerosol droplets. And that's similar to how coronavirus is being transmitted. We are part of the Flying Scientist program, and typically our program flies and sometimes even drives scientists from very different areas of expertise across Queensland. We visit remote and regional schools and communities in order to bring all the exciting research that we're doing to you. Unfortunately, at the moment, due to the unfolding situation with coronavirus and the restrictions around travel, we are unable to visit.
Today we are social distancing, so I'm here on the Sunshine Coast.
And I am all the way up in Cairns in my office at JCU.
So we are just 2 of 40 scientists involved in the Flying Scientist program. You'll hear from a couple more scientists at the end of this video, but today we're going to present the first in a series of videos by Queensland scientists discussing their cutting-edge research and areas of expertise. So, Dr. Andreas and I are going to talk about our areas of expertise, viruses and infectious diseases.
Obviously, viruses are a big cause of concern at the moment, but what exactly is a virus? In contrast to bacteria, which are very, very small living organisms, a virus is an even smaller infectious agent that requires the cells of living organisms to replicate. And when a virus infects a cell, it uses this cell to make thousands, sometimes millions of identical copies of itself. Virus can infect almost all organisms, including bacteria. Those viruses that infect bacteria are called bacteriophages. But there's also many viruses that infect humans and cause diseases such as diarrhea, AIDS, and COVID-19.
As microbiologists, there's lots for us to learn about viruses. We run experiments to understand how viruses spread between people and animals and how different viruses make us sick in different ways. We also work to find medicines and drugs to slow down viruses replicating or copying themselves in our cells and work to develop vaccines to help our immune system fight back. So, why have so many of us been staying at home over the past few weeks? Because we want to reduce the transmission of coronavirus. Well, do we know how viruses transmit or spread? All viruses are a little different, but overall, viruses spread in 3 major ways: direct contact, indirect contact, and airborne by aerosols. To understand how viruses spread in these ways, let's do an experiment. Let's pretend we are a new virus and see how far we can spread by these 3 ways. To start, either on your own or in teams, decide on a name for your new virus. I'll test virus Spartacus. Your virus could be Magic Unicorn or ABC123 or whatever you like. For each way to spread, or mode of transmission, we will do an experiment and record the results. To measure direct contact transmission, you will need a measuring tape. Pick a starting mark on the floor and, keeping your feet behind the mark, reach out with your hands along the floor as far as you can. Measure the distance from the starting mark to the end of the farthest part of your hand. Don't let any other body part touch the floor, just your feet and hands, like you were doing a push-up. Record the distance you reached. If you are a team, take the average of each team member's distance. to measure indirect contact transmission, you will need sticky notes and a timer. You can use plain paper and tape if you don't have sticky notes, and you can use a regular watch to keep track of time if you don't have a timer. If you are doing the experiment with multiple teams, make sure to either have different colored sticky notes or to mark your notes with some kind of identification for your virus. I'll put an S on mine for virus Spartacus. Then, with your notes, have someone time out 20 seconds. In that 20 seconds, you and your team need to place as many sticky notes around the room as you can. Put them on all the things you would touch in the room. After the time, count up how many notes you stuck and record the number. Finally, to measure airborne transmission, you'll need your measuring tape and timer again, plus you'll need bubble solution. So check whether you could do this activity inside or outside first, then pick a starting mark on the ground. You're going to start by blowing bubbles for 10 seconds. After 10 seconds, start your timer and record how long it takes for all the bubbles to pop. As well, keep an eye out for how far the bubbles travel and take your measuring tape and measure the distance from the starting mark to where the farthest bubble pops. Record both the bubble time and distance. Got it? Okay, you go measure your new virus and I'll go measure virus Spartacus and we'll meet back here. Welcome back. How did your experiments go? For me, I found that virus Spartacus could travel 141 centimeters by direct contact. That's just over 1.4 meters. Was that closer or farther away than your virus? Now, direct contact transmission is spreading a virus by directly touching someone. So, knowing that, how might we stop our virus from spreading by direct contact? The two big ways we stop direct contact spread is by washing our hands to remove as much virus from our hands as possible, and by staying 1.5 meters apart so we can't touch anyone. Sound familiar? Okay, how did your virus do for indirect contact? How many items were you able to tag with your notes? Virus Spartacus tagged 12 items in 20 seconds. Now, indirect contact transmission is leaving the virus behind on things we touch for other people to touch once we've gone. We call items that hold viruses like this fomites. Now, how do you think we could stop indirect spread of a virus? It's very similar to direct spread. We still wash our hands to have less virus on our hands to leave behind. And we also clean surfaces with good cleaners and disinfectants to remove virus we may have left behind. Finally, viruses can also spread in the air on little droplets of water we breathe out called aerosols, which are very similar to tiny bubbles. So from our experiment, how long did your virus bubbles last and how far did they travel? Virus Spartacus lasted 27 seconds and traveled 920 centimeters, or just under 10 meters. So airborne transmission can be really hard to stop. How would you try and contain your virus from spreading in the air? To slow down airborne spread of a virus, one of the best things we can do is stay at home if we're feeling unwell. This keeps the virus in our house and not all over the neighborhood and in the shops. If we need to go out when we're not feeling well, it helps to wear a mask. A mask stops the water droplets we breathe out from traveling away from us. It would be like trying to blow bubbles from behind a mask. The mask would catch the bubbles before they can drift away. So I hope this set of experiments has helped you understand how viruses spread and why we're doing the things we are doing to stop COVID-19 from spreading.
So now we know how viruses spread, but how do we stop them or slow down their transmission? Over the last few weeks, you may have heard the terms herd immunity and vaccines. Do you know what herd immunity and vaccines are? If you do, that's great. But if not, that's okay too, because today I'm going to tell you why both of these things are very important in our fight against an infectious disease. such as COVID-19. In contrast to drugs that we can use to treat people when they are sick from an infectious disease, a vaccine is used to prevent the infection in the first place. What a vaccine does, it trains or primes our immune system so that the body knows how to fight the virus or bacterium. If we then get exposed to the same pathogen again, the body knows how to eliminate it and we don't get sick. For many diseases, including COVID-19, there's no vaccine available yet. Fortunately, we know that many people that have been infected with a virus or bacterium and get sick usually recover after a while. For many of them, it also means that after recovery, they are immune to the disease. Being immune means they cannot get sick again and are protected. This is very similar to what happens after vaccination. To illustrate the importance of vaccination and herd immunity, today I'm going to show you a game that you can play at your school or at home. For this game, you can pretend to be the virus. The game will teach you how important it is for people to be immune in order to slow down the spread of a virus. All you need for this game are a few buckets and a set of balls, ideally of 2 different colors. Today I'm using orange and white table tennis balls. An orange ball represents a person that has not been vaccinated and has not been infected before. So these are susceptible individuals that you can infect as the virus. A white ball represents, however, a person that has either been vaccinated or has already recovered from the infection. So these are immune. Rule number 1, you're not allowed to look inside the bucket when you grab a ball. Rule number 2, when you grab an orange ball, you can take it out and put it aside. Rule number 3, if you grab a white ball, you have to put it back because this ball is immune and you can't infect it. Number 4, you measure the time, how long it takes until you have infected all orange balls. And number 5, you also count the number of times it takes you before you infected all orange balls. Okay, in bucket number 1, you can put 20 orange balls, but no white balls. Then you grab in the bucket and see how quickly you can infect all orange balls. What do you think? How long will it take? Bucket number 2. In this second bucket, you need to put 10 orange balls and 10 white balls. This means that half of the balls, or 50%, are immune. So now you play the game again and see how long it takes you to infect the remaining 10 orange balls. What do you think will happen? Will it be faster for you to infect those 10 orange balls, or will it take longer? In the third bucket, we can put 18 white balls, but only 2 orange balls. This means that 90% of all the balls are already immune and you can't infect them anymore. So now play the game and try to see how long it will take you to infect the last remaining 2 orange balls. What do you think? Will it be faster again? To illustrate how the game is being played, I've chosen bucket number 2. Bucket number 2 contains 10 orange balls and 10 white balls. So remember, we don't look. You grab into the ball bucket, grab a ball, orange ball. I can infect it, so I can take it out. Do it again. White ball, immune, I can't infect it, I have to put it back. Orange ball, can infect it, take it out. And so on. And you play the game until you've been able to infect all remaining 10 orange balls. So what were your results like? I suspect for bucket number 1, your results will be very similar to mine. Why is this? Oh, because every single ball in bucket number 1 was orange, and every time you got into the bucket, you could infect the ball. That means the virus can rapidly infect the entire population. And that's very similar to what's happening at the moment with coronavirus. No one is immune, we don't have a vaccine, So every person the virus comes in contact with, it can infect. And that's why we all have to stay home at the moment, to slow down the spread of COVID-19. And what happened in bucket number 2? Did you find it took you longer to find the balls, or was it faster? You can see my results were longer than bucket number 1. And this is because the likelihood of us, the virus, finding a person that we can infect is much lower, because half the population is already immune. And how did you go with bucket number 3? Did you manage to infect the last 2 remaining orange balls? You can see, for me, it took more than 3 minutes to find the 2 balls that I could infect. If you were lucky, it was faster for you, but even if you managed to infect those 2 remaining balls relatively quickly, there were no other balls left for you to infect. So, in this game, we have explored the concepts of herd immunity and vaccination. It illustrates the importance to develop a vaccine for an infectious disease, and it shows that the more people are vaccinated or have recovered from the infection, the more difficult it is for the virus to spread. And this is why so many scientists around the world are currently trying to develop a vaccine for coronavirus.
So, I hope Dr. Andreas and I have been able to help you understand a little more about viruses, how they spread, what we can do to slow them down, and how vaccines and immunity can help you stay healthy.
Today you have seen that scientists are very important in fighting infectious diseases and to come up with solutions. Microbiologists around the world are working on many infectious diseases, and COVID-19 is only one example of a disease for which we will need a vaccine to stop it. Given that so many groups around the world are currently trying to develop a coronavirus vaccine, I'm very hopeful that soon we will see some positive results. And remember, the more people are vaccinated against the disease, the more likely we are to eradicate it. This has happened before. Smallpox is an example of a disease which has been eradicated because an effective vaccine was introduced.
Thank you so much for watching our first Flying Scientist video. But what next? Well, we want to hear from you. Now let's hear from some of our other flying scientists about how you at home or at school can be involved in our future videos.
G'day, my name's Ben Allen from the University of Southern Queensland, Toowoomba. I'm a wildlife research scientist where I have the best job in the world. I do science called wildlife management, but all of the science that I get to do is related to animals and managing animals. It's really important for our farmers, for our national parks, for our native animals, and for all the people who enjoy this wonderful country. You get to do some weird and wonderful things, and hopefully we'll get to do them for a long time more.
Hi, I'm Dr. Michaela Mitchell, and I'm a marine biologist, but I'm a very specialised marine biologist, and I study sea anemones. I am a taxonomist, and I name and describe them and identify them. But I'm also studying their venom so that we can use their toxins in medicines to treat diseases like diabetes and multiple sclerosis.
Hi everybody, my name's Paul Salmon and I'm a Professor of Human Factors at the University of the Sunshine Coast. My research is all about understanding and optimising the behaviour of complex work and societal systems, and it can take on many different forms. This might include accident analysis and investigation to better prevent future road, rail and aviation crashes, designing or redesigning work systems to make them safer or to make them more efficient, and designing things like robots so they work well with their human teammates. Ultimately, the overall aim of all of my research is to enhance human health and well-being.
If you have a burning question you'd like answered,
Or a particular scientific topic that you'd like a scientist to speak more about,
please send it to us at Wonder of Science
by the 22nd of June, and we'll organise someone to prepare a response for you at one of the National Science Week events.
Keep an eye on the website for details of those events. And remember, you're never too young or too old to wonder and to want to know more about science and the amazing scientific research that's happening right here in Queensland.